I'd like to thank the organizers for letting me give a talk on such short notice. As introduced, I'm a fifth-year grad student at Vanderbilt, and I've been working as an intern in the Data2 Foundation, and I'm excited to share some really hot data regarding the insights that we can glean from alpha missense in the context of ADA2. So I'm sure everyone can agree that the disease mechanism is yet unclear. We've heard that over and over again, and that the heterogeneous clinical manifestations make it really difficult to make connections between genotype and phenotype. So I've listed some of the potential mechanisms of disease here. I'd like to focus on the last one, where the adenosine regulation, deaminase activity of ADA2. So it's been established that some variants that have residual ADA2 activity, mostly they present with vasculitis, and the more deleterious variants like missense or nonsense and deletion, frame shifts, they result in blood disorders and immune deficiencies. And needless to say, patients present with very low ADA2 activity compared to healthy controls. And this could be due to a complete lack of ADA2 in the bloodstream, or it could be that the enzyme itself is non-functional. All right, so I started out by asking a question. So are there any structural hotspots where all these variants sort of cluster around and hit the enzymatic activity the hardest? So I started out by mapping out these variants onto the protein sequence. A lot of these came from this excellent review by Zuz et al., so definitely a shout out. I've also added a bunch of unknown clinical significance variants discovered from in vitro genetic screening. So these are unknown clinical significance. And then I've also added some variants that were provided by the Data2 Foundation, our own tier 0 patient cohort that are highlighted in green. And so do let me know if you don't see your favorite variant on here so that I can add them to this. So just overall, I have about 96 missense variants listed here. And then on my poster, there's additional total 170, including all the intron, exon, and frameshift mutations. So it's really pertinent that we should continue to expand this database of variants. All right. So what are these? What are the hotspots? So going back to my first question, I've sort of highlighted with boxes some hotspots that are being hit. So for instance, in the dimerization domain, where it includes one of the founder variants, as well as these 4 red boxes in the catalytic domain. And these, if you fold the protein into a 3D structure, happen to constitute the active sites. So it seems that the catalytic activity is being hit, and that the activity may be important for disease phenotypes. Right. And so if I map this out onto a 3D structure, you get something like this. The red variants are marked in red onto a 3D structure. This is a crystal structure. And the monomeric form is shown here with the domains as colored so. And then you can already see that there's a hotspot that begins in the interface between the dimerization domain and the catalytic domain, likely important in forming a dimer and leading to activity, but that can be lost with these variants. And then this ribbon representation, you can also see that the variants are sort of peppered around or clustered around the active site, likely leading to a loss of function. And in these different viewpoints or angles, you can already also glean that the hotspots are located on the catalytic domain, likely leading to some sort of recognition by protein folding machinery or degradation machinery, or some direct defect introduced to the catalytic activity. And so where am I going with this? So alpha missense got published 2 weeks ago. It's a really new algorithm. So basically what they did was they leveraged AlphaFold, so its ability to predict protein structures, and then combined it with evolutionary conservation data. And so you throw in an input of protein sequence, and then you change one of the amino acids into a random one, or a different one, which is a missense variant. And then you go through these algorithms, which then predicts and spits out an output, which is a number that ranges from 0 to 1. Anything above 0.564 is considered pathogenic based on their cutoff. Anything below 0.34 is considered benign. And they did this for all the proteins in the human proteome, so every single possibility. Huge data set, so of course 882 was in here. So I went ahead and started to sort of see what we can glean from that information, what kind of missense variants, what predictions were made. All right, but before we dive into the actual data, I'd like to just showcase a benchmarking that we did. Actually, this was done by Eli McDonald, a fellow grad student in the lab. So our lab sort of studies CFTR and cystic fibrosis. It's a well-documented disease. There's many clinical outcomes, patient data, in vitro data that we can use to benchmark this algorithm. So right off the bat, you can see that this is a RSC curve. The alpha missense is pretty good at predicting benign variants, as well as pathogenic variants. It's not so good at predicting unknown clinical significance, unsure about the ambiguous variants. If we correlate this with in vitro data, so channel activity is a readout of pathogenicity for cystic fibrosis. Here we're mapping about 800 variants that are expressed in FRT cells, and we're correlating that with the alpha missense score for each variant. So higher the score, again, the more pathogenic it is. So you'd expect lower function with a higher score, so there's negative correlation. And then we can also look at patient data. So for instance, sweat chloride is a measure of disease severity in CF. Patients get salty sweat, so more salt in their sweat, it would indicate higher disease severity. So it gives a nice positive correlation in this case. There are other metrics that we also looked at. I'm just showing one of them. So with this in hand, now we move on to looking at 882. All right, so the code for making these plots and doing the analysis was provided by Jake Hermanson in the lab, another fellow grad student. So what I'm showing here is a dot plot of every single residue on 882, so from 1 to 511. And each column has a score for the 19 different amino acids that can be substituted. So it's an extensive, exhaustive data set. And again, anything above this red line is considered pathogenic, and anything below this blue line is considered benign. And if we sort of match this with the variant map that I've made with the available clinical data, you can already see that some of these hotspots that were seen in the catalytic domain match up with these red dot clusters that are around. And then if we look at it in the density plot, again, the high density positions sort of match with the catalytic domain hotspots here, here, and here. And also interestingly, there are cold spots where the amino acids are pretty resistant to any changes that are introduced. For instance, in the signal sequence, which I can talk more about if you're interested in talking about this. It's likely misinterpreting the signal sequence variations. However, in the putative receptor binding domain, there's a cold spot. And in the empty areas where we don't have any clinical variants known, it actually predicts it as benign. So that's pretty interesting. Alpha missense seems to be pretty good at predicting these general hotspots within the protein. And so if I take the average of each residue, 19 scores within each residue, we get something like this. Sort of hard to see, but basically the takeaway is that the big red columns would indicate the hotspots that are the most vulnerable and are most conserved, basically. And when perturbed, introduces a structural defect that would likely lead to loss of function. All right, but this is all in silico data. So let's look at some actual functional data. Well, actually, going back, when I mapped out the average scores, it looks like this onto the protein. So a lot of the outside exposed residues are pretty tolerant to changes, whereas the central amino acid that constitute the active site are hit the hardest, which sort of makes sense. And so let's look at the in vitro ADA2 activity data as published by Dr. Lee. These are all the variants that had available ADA2 activity, at least in vitro. And they are all considered pathogenic or ADA2 or DADA2 related, although they do show some variable activity levels. And so if I put alpha missense predictions right below it, you can already see that it's getting some of these wrong. Like these all should be theoretically pathogenic if predicted correctly. But what's interesting is that it actually predicts the residual activities pretty well in the sense that it predicts it as either ambiguous or benign. And so what it seems to be doing is that it can actually predict derive a structural defect into an activity profile. So if you looked at in a continuous scale instead of a discrete scale, on the y-axis, ADA2 activity is shown for all these variants. On the x-axis, we have the alpha missense score. So again, higher score is more pathogenic. We get this negative correlation as expected, but then I noticed that there's some sort of a bimodal distribution where there's a group of variants that nicely follow that proportional decrease as its structure is more compromised with these defects, which would then be the structural defect induced by a decrease in function. But then there's also these variants that just sit on the bottom, which it seems to me that these are being predicted as little to no change in overall structure, but in reality, they result in no effect, no activity. So it seems that these are caused by some structure-independent loss of function, likely due to some other protein interactor-derived activity loss, maybe like degradation, or maybe like DNA binding. All right, so I want to circle back to these variant phenotype issues. So like I said, the missense variants sort of show vasculitis. The other ones show different phenotypes. And so we should actually start looking at these variants separately, like group them by their phenotype, for instance. So as part of that effort, with help from Julie Williams at the DADA2 Foundation, we started categorizing these variants into each big phenotypic category. And now we can do independent analyses within each category instead of doing an aggregate analysis that will somewhat skew the results. And so we should try to move towards that. And then one last thing I would like to point out, that alpha missense could be used as a diagnostic tool really quickly. So for instance, these variants that I've listed here, W123C and R169W, these are some variants of unknown clinical significance as reported by Invitae from their genetic screen. And I looked them up in the database, and lo and behold, they are pathogenic. So it seems that it could be helpful. You can make some informed decisions in predicting the pathogenicity of variants that are unknown and are rare, and especially in the context of heterozygous patients. All right, so looking forward, these are some ideas I had while I was making these slides. So like I said, we should start looking at disease severity in the context of disease severity and phenotypes, and then maybe the prediction scores, correlate them also, and then start getting some unified metrics that could, that in addition to the ADA2 activities, that we get a better sense of what kind of disease metrics can be correlated to the scores. All right. And then it seems to me that it's really not well understood what the exact protein homeostasis machineries or interacting protein profiles are involved throughout the biogenesis of ADA2. And so it'd be really important to know that, because then you can start targeting the various proteostasis factors that can maybe help fully traffic or make functional ADA2. And so if we do that, then we can start annotating the mutants according to their defect. For instance, these are some just random classes that I threw up. So class 1, for instance, complete loss of protein resulting from nonsense frame shift. And then class 2, lack of protein, or resident in the ER due to misfolding or mistrafficking. And then some class 3 mutations that have lack of function, namely the T360A and G383S, which are actually traffic and get secreted but have no activity. So some conclusions. Hopefully I've convinced you that there are some hotspots on the protein that are in the dimerization and catalytic domains, and that alpha missense can be used to predict the pathogenicity of these missense variants. These are some strengths and limitations that I listed. Alpha missense seems to be pretty good at predicting protein function because it uses its structure and function follows form. However, it's only limited to single missense mutations, and it doesn't seem to be performing well for predicting other factors that are involved in protein processing. Right, and so again, our analysis is somewhat limited by the availability of a wide variety of clinical data sets. So it'd be nice to have those, and then they'll definitely further advance our understanding. And with that, I'll be thank you for listening, I guess.